Hello, and welcome to this detailed biology practical session, which is brought to you by a senior KCSE examiner. In today's video, we'll walk step by step through 3 commonly tested practical questions, from identifying leaf adaptations and calculating surface area, to conducting food tests and using a dichotomous key to classify specimens. Whether you are a candidate preparing for your exams or a teacher guiding students, the video will sharpen your skills and improve your practical confidence. Let's get started. As we normally do in this 1996 paper 2, biological sciences practical, I'll start with the confidentials. And here is what was required for question 1. So, question 1, confidential. A freshly picked bougainvillea leaf labeled specimen K1, a razor-blade or scalpel, a freshly picked leaf of kale, skumawiki, locally named, labeled K2, a petri dish, methylene blue or 1% methylene blue, microscope slides, or you can call them grass slides, and a hand lens. So, I'll first of all show you the specimens for question 1, and those are the bougainvillea leaves. And this is my work. So, the other specimen was to be got from skumawiki or kale, and you can see the skumawiki there. I think you can see it here. This is skumawiki, this one. Because I took the photograph in my kitchen garden. The others here are beans and amaranthus hybridus, but this is the skumawiki. And there you have the question 1 specimens. So the leaf of bougainvillea was labeled K1, and the leaf of the kale was labeled K2. And I photographed this myself. Let's now go to the combinations of question 2, because we are through with question 1. In question 2, you are needed to have a groundnut seed which had been soaked in water for 24 hours, and it was labeled M1. Then you had to have a half teaspoonful of groundnut powder crushed using pestle and mortar, labeled specimen M2. Remember, you are the one to crush in the laboratory. You have also iodine solution. Then you have dilute sodium hydroxide, NaOH, 1% copper sulfate solution, Benedict solution, white tile, Bunsen burner or means of heating, 2 test tubes, and a test tube rack. So I went to Wikipedia and I got this photograph of a groundnut plant. And of course, in YouTube, you are supposed to take care of copyright. And I found that this was in the public domain. Specimen M or groundnut seed, which is now going to be pounded to get specimen M2. So I did not do the pounder, but for those who are going to attempt this practical, please you'll be required to get M2, which is the ground flour of groundnut seed. Finally, as far as confidentials are concerned, we come to question 3, where you are needed to have a beetle with all parts intact, labeled specimen P, another worm labeled specimen Q, and a wingless ant labeled specimen R. And there you are. You have specimen P, specimen Q, and specimen R. I would do not good if I don't give the attribution, and I gave the one for the ladybird, all right. Then I gave the other one for land Californian earthworm, and then we have the weaver ant in fighting position, and therefore I think that one takes care of my copyright accusations. So in question 1, you are provided with the specimens labeled K1 and K2 as we saw. You are given this graph. Use the graph paper provided to determine the surface area of specimen K1. So you give the surface area there. So let's move very fast through the questions. 1A2 is you describe how you arrived at your answer. B, what is the significance of surface area of the leaf to the plant? How is the specimen K1 adapted to its function? That is C. D, using a laser blade, make the intersections of the pesher of specimen K2. That is the kale. Place the sections in a petri dish containing methylene blue. Allow to stand for about 1 minute. Select the thinnest section and mount on a glass slide. Observe the section using a hand lens. Roman 1, you are going to make a labeled plan diagram of the section. Roman 2, what was the purpose of using the methylene blue? And Roman 3, state the functions of any 3 structures which you have labeled in D1 above. And finally, E, from your observations of the section only, what evidence is there to show that the specimen is obtained from a dichotomous plant? Question 2. You are provided with specimens labeled M1 and M2. Split specimen M1 open. A, draw the inner surfaces side by side. Label it. B, work out the magnification of your drawing. 1 mark. And C, select and use the reagents and materials provided to test for the presence of starch and proteins in M2. Record the procedures, observations, and conclusions in the table below. So the table is there. Food substance, procedure, observation, and conclusion. The food substance is starch and protein. You are provided with 3 specimens labeled P, Q, and R. Use the dichotomous key given below to identify the specimens. For each specimen, write down the steps you followed to arrive at your answer. Sorry for that, guys. So you have the dichotomous key there with 1 all the way to 6, the one you are going to use. Remember, we have already attempted this in the biology practicals in my videos. Please don't forget to watch the other biology practical videos from the channel Smart Traveler Memos. I'm a traveler and I'm a tutorial fellow. Then we have the 7, 8, and 9 to clear the dichotomous key. So specimen PQR, identity, steps forward. You clear with your question 3. So now let's enter the kinds of marking scheme. For question 1, Roman 1, you have your drawing there. That is the way they expected you to come with. And the surface area will be equal to, you are supposed to give units in square centimeters or millimeters. All right? Because this one is a squared paper. And you can see we are going to explain what is going on. So that is the way you did your leaf, by tracing. But we will tell you. Neck was marking the following. You are supposed to describe how you arrived at your answer. So, the first mark, you have to have evidence of tracing. You have to mention tracing. Number 2, you should show the counting of squares. Number 3, you multiply by 2. And number 4, divide by 4. To get square centimeter, if answer is in centimeter. 4 marks, maximum 3 marks. So, because the kinds of marking scheme was brief, I went to ChatGPT. So, this is exactly what you did. Trace the outline by doing the following. Place the Bougainvillea leaf specimen K1 flat on the graph paper. Carefully trace its exact outline with a sharp pencil. That's what you do first. Count full squares. That is, you count all the full 1 centimeter squared squares within the outline. Each full square is equal to 1 centimeter squared. Next, you estimate partial squares. For squares partially filled by the leaf outline, combine 2 halves or 4 quarters to make a full square. Count how many full equivalents you get. Add these to the full squares. Finally, add them up. The total surface area equals full squares. Sample format here, you are going to fill your own value. So surface area calculation is full squares 24. So there you fill your whatever you got. Partial squares estimated full equivalents were 6. Therefore, the total surface area of K1 is 24 plus 6, which gives you 30 centimeters squared. Quite good. Let's go on. What is the significance of the surface area of the leaf to the plant? According to the NEC, or the Examination Council Marking Scheme, the significance is photosynthesis. So if you say into photosynthesis, you get a tick. Gaseous exchange, you got a tick. Transpiration, you got a tick. And large surface area is more efficient. If one efficiency occurs, I want to mark only once. Efficiency, a lot, increase, quick. Those are alternatives. For the ChatGPT, the significance was even explained better, so that we have number one is photosynthesis, the significance of the surface area. A large surface area allows the leaf to absorb more sunlight, enhancing the rate of photosynthesis for food production. So you see now for ChatGPT, we are explaining gas exchange. A broad surface facilitates efficient exchange of gases. Carbon dioxide in, oxygen out through stomata. Number 3, transpiration. The large surface area increases water loss through transpiration, which helps in cooling the plant and creating a transpiration pool for water uptake. 4, absorption of carbon dioxide. A wide leaf surface provides more space for carbon dioxide absorption, essential for photosynthesis. Remember, in the present naming, we normally call carbon dioxide carbon 4 oxide. Carbon dioxide is carbon 4 oxide. How specimen K is adapted to its function, Council Marking Scheme. It is thin, flat, large, broad surface area for gaseous exchange, transpiration or photosynthesis. Any of those ones. Remember, guys, I told you don't put strokes in marking because the council will penalize you, will think you don't know what you are saying. So you pick only one of the ones in the stroke. For example, I can pick thin, broad surface area for gaseous, thin for gaseous exchange, or thin for transpiration, or thin for photosynthesis. Then broad surface area for gaseous exchange, broad surface area for transpiration, broad surface area for photosynthesis. Veins for transport, support, petio, midrib for transport, support, petio for attachment. Guys, take care. Don't put in a stroke there. I have indicated the strokes to expand and look at all the alternatives which you can put. Chlorophyll or green color for photosynthesis. Those were the adaptations. Let's go to ChatGPT. I don't know whether we have it. Let's see. Yes. Broad surface area, it decreases the surface exposed to sunlight, enhancing the rate of photosynthesis. Thin structure minimizes the distance for diffusion of gases, allowing efficient gas exchange. Presence of stomata facilitates gaseous exchange and transpiration, aiding in cooling and nutrient transport. There are instructions here, like the ones in red. If phloem and xylem is labeled, it must be indicated properly and bracketed. No marks for drawing because it is a plan. So here we did not have a drawing mark because you had been told to draw a plan. So you can see there are various shapes. The one up there is the general one where you have the pith, the epidermis, the cortex, and the vascular bundle. Notice that you cannot say phloem and xylem. They cannot be seen. So if somebody labels phloem or xylem, we cancel. Let's go to the other diagrams here. Again, you can see we are concentrating on 4 labels: pith, vascular bundle, epidermis, and cortex. There are even some who drew a longitudinal section where you have epidermis, cortex, vascular bundle, pith. Okay, 4 labels, 4 marks. So there was transverse section and longitudinal section, which the council looked at. So here, the instructions are: accept diagrams both in single line and double line. If phloem and xylem is labeled, it must be indicated properly and bracketed. No marks for drawing because it is a plan. Allow label xylem, phloem, or vascular bundle provided correctly drawn. If there are strokes, xylem, phloem, vascular bundle, you can allow. Interchange of phloem and xylem give for functions. Labels to include in your drawing. You have to put epidermis, you have to put pith, cortex, or parenchyma, which is supporting tissue just below the epidermis. Then there was the vascular bundle or central region containing xylem and phloem. But they cannot be seen. These ones cannot be seen. Question 1D. What was the purpose of using methylene blue? To stain the tissues, or to make the structures more visible or more clearer, or want to that effect. State the functions of 3 structures which you have labeled in the above. We have epidermis for protection, cortex for strengthening and support, vascular bundle for transport, food and water, pith for feeding or packing or storage or strengthening, phloem for transport of food, xylem for transport of water and mineral salts. E. From your observation of the section only, what evidence is there to show that the specimen is obtained from a dichotomous plant? Is that vascular bundles are arranged in a ring or circular. And for ChatGPT, epidermis protects inner tissues from mechanical damage and reduces water loss. Parenchyma or cortex provides mechanical support and flexibility to the tissue, helping it to withstand bedding. Vascular bundle for ChatGPT broke it to xylem, transports water and mineral salts from roots to leaves, and phloem transports manufactured food from the leaves to the other parts of the plant. Evidence that the specimen is from the dichotomous plant, the vascular bundles are arranged in a ring, which is a characteristic feature of dichotomous plants. Question 2. Draw the inner surfaces side by side and label it. You can see specimen M, four labels, maximum 3. We have the tester, we have the cotyledon, we have the plumule or first forage leaves, and we have the radicle. 4 labels drawing. So tester should be differentiated from the cotyledon. There must be a double line. Orientation, radical towards the outside and plumule towards the inside. Oh, there is a drawing mark, sorry guys, because there are 4 labels which are giving you maximum 3 marks and 1 mark for the drawing, and the total mark is 4. Continuity of lines must be proportional. Reject if there is any shading or if only one cotyledon. Labels include cotyledon, plumule, radical, seed coat for ChatGPT. So guys, there was a problem. My video was blinking, but now I'm trying to stabilize. So we were through with that. That is the work of the working out the magnification of the drawing. And I said measurement of the drawing or measurement of object. And the council requires you to have times 1 as the magnification. So we can explain. To calculate the magnification, use the formula magnification is equal to size of drawing over actual size of specimen. Steps to work out magnification: measure the length of the drawing, e.g. from the tip of the radical to the top of the plumule in centimeters. For example, you get 8.0 centimeters. Measure the actual size of the groundnut seed, specimen M1, in centimeters or millimeters. For example, 2 centimeters. Plug into the formula magnification equals 8.0 centimeters over 2.0 centimeters. The centimeters will cancel and you will be left with 4 times. Or you can put the X in front or at the back. Final answer format is magnification is equal to 4 times. So here you replace your actual measurements. Explanation from GPT. Now, selecting and using the reagents and materials provided to test for the presence of starch and proteins in them too, you record the procedures, observations, and conclusions in the table. And you have food substance starch. Procedure is you add a drop of iodine solution. There are 2 procedures there. You get blue-black or dark blue color if starch present. The other procedure you could have used was you put M2 on a white tile and you add iodine solution. You get a blue-black color and you say starch is present. So you could either use a tile or a test tube. Protein, you add sodium hydroxide and then followed by copper sulfate solution. Purple color or violet color, protein present. For the ChatGPT, this is what I found out. You add a small amount of M2 to a test tube and a few drops of iodine solution, shake gently. Color changes from brown to blue-black, starch present. protein and a small amount of M2 in the test tube, and burette reagent or a mixture of sodium hydroxide and copper sulfate. Shake gently. Color changes from blue to purple or violet. Protein is present. That is the way the ChatGPT was explaining. And we are through with question 1. We come to question 3. And here you are identifying P, Q, and R. And the identities were P was Coreoptera. Steps followed are 1A, 2A, 3A, 4A, 7B, 8A. Remember, each of these steps is a mark. That's why you are seeing semicolon. Q is an reader, 1A, 2B, 5A, 6A. And R is Hymenoptera, which is 1A, 2A, 3A, 4B, 9A. You can go back to the dichotomous key and see those steps, and you come to the identity. So you are getting 18 marks. You divide by 2, you get 9 marks as your marks. To identify specimens P, Q, and R, according to ChatGPT, using the given dichotomous key, you need to observe their features like body segmentation, presence of appendages, number of legs, wings, body shape, etc. Tips for full 9 marks: use correct terminology. Clearly follow and list each dichotomous step. Match observable features to each branching point. So, my closing remarks are as follows. Thank you for watching. I hope this session has helped you understand how to tackle KCSE Biology practical questions with clarity and confidence. Remember, mastering practical is not just about memory. It's about understanding the steps, making accurate observations, and linking theory to real-life specimens. Don't forget to subscribe for more videos like this and leave a comment if you would like us to cover a specific topic next. Best of luck in your studies and upcoming KCSE examinations. So we acknowledge by saying that we wish to sincerely thank the Kenyan National Examination Council for providing the foundational materials that guided this marking procedure. Special appreciation goes to ChatGPT, whose support in structuring and refining this video content made a big difference in presentation and clarity. I'm deeply grateful to my colleague examiners who contributed their insights during the moderation sessions. Your collaboration enriched the depth of this story. Finally, heartfelt thanks to my family for their unwavering support and patience during the long hours I spent preparing the PowerPoint presentation and recording this video. Your encouragement made this possible. See you guys, and goodbye.